Good evening, everybody. I would like to begin my express gratitude to Alex and his team for their commendable effort towards the advancing the field of toxicology in India. It's my pleasure welcome to you all. Thank you for joining today. My presentation is on nanotoxicology, where I am covering nanomaterials and their toxicology aspects. The objective to talk provide a clear understanding of nanotoxicology, and I hope to share insight that will particularly valuable in context of DABT examination. So first of all, what are the nanoparticles? The nanoparticles are the ultra small material which dimension between 1 to 100 nanometers. 1 nanometers means 1 billionth of meters. At this scale, nanomaterials show unique physical, chemical and biological property that differ from their bulk form. Some of the important features include like high surface area to volume ratio. This increases their reactivity and ability to interact with the biological system and other material. Enhanced reactivity because of so many atoms are exposed on the surface. Reaction happen more rapidly than the bulk material. They have tunable properties at the nanoscale. Properties like color, magnetic conductivity, even mechanical strength can control simply by changing particle size, shape and surface chemistry. Because of this property, they are widely used different field you can imagine. In the medicine, they are used drug delivery system, imaging, diagnosis. In electronics, they can help in develop smaller, faster, more efficient device. In the energy sector, they improve batteries, fuel cells, solar panels. They also use in cosmetic as a sunscreen and anti-aging cream. In environment application such as water purification and pollution control. When we talk about the engineering nanomaterials like nanosilvers, it is widely used for the antimicrobial property. Fluorines, it is used as a carbon-based nanostructure with unique strength and electric property. Quantum dots, the semiconductor nanoparticles, they have remarkable optical property useful in imaging and display technology. Carbon nanotubes, also known as exceptional strength and conductivity. Graphene based material famous for being strong, light and conductivity. Metal oxide nanoparticles such as titanium oxide, zinc oxide, commonly used sunscreen, coating and catalysis. So overall, all the nanomaterials have unique class of material with property to make them both powerful tools and important subjects of nanotoxicology. So there is a different types of nanoparticles in classes 9th edition, page number 1982. There are starting from like materials, different types of material like sodium, fructose, protein, virus, bacteria, even baseball. You can see how the nanoparticles fall in the same size range as a virus and protein, which can explain why they so easily interact with the biological system. As bottom, we can see a different nanostructure for bulky ball, fluorines, carbon nanotubes, graphene sheets, zinc oxide, nanorods and nanonears, all which engineered nanomaterials with distinct structure and distinct property. On the ultrafine and nanoparticles, natural and anthropogenic source, two types of nanoparticles, natural and anthropogenic. In natural and anthropogenic is intentionally and unintentionally. So in natural source include process like volcano eruptions, forest fires, gas to particle conversion, and the biological origins such as virus, magnetic, bacteria. Unintentional anthropogenic source, mainly byproduct of human activity, such as emission from the internal combustion engine, power plant, airplane jets, smelting, welding, polymers fume, and they even cooking process like frying and grilling. Intentionally anthropomorphic zones like well engineering nanoparticles, they are significantly designed for control size, shape, surface chemistry for desired function. Example like metals, semiconductor, carbon-based nanomaterials, metal oxides, which is applied in cosmetic, medicine, electronics, optics, and display. So overall, nanoparticles occur naturally, unintentionally from human activity, and intentionally from the engineer, regardless of source. Nanoscale size, unique property make highly relevant in toxicology and safety study. There are different types of properties of nanoparticles. First of all, high surface area and reactivity. That larger particle that compared to the larger particle, nanoparticles expose many more atoms on their surface that make high reactivity, enhance their ability to absorb chemical, lead to the strong catalytic activity. It also increase their dissolution rate, which can directly influence both application and their potential toxicity. Next, surface chemistry and morphology play key important roles to determining how interact with the biological system. Factors such as surface coating, impurities, even the shapes or curvature of the particle affect how the dissolve, release ion, behave like fluids like blood and lung secretion. This is why two nanoparticles of the same material can behave very differently in their surface structure, not identical. Also, they have unique electronics and magnetic property. Nanoparticles can exhibit unique electronics and magnetic property, such as their tunable fluorines in quantum dots or super paramagnetic in paramagnetic nanoparticles. This property can exploit in various technology applications. In this summary, the high surface area, surface chemistry, and unique electronics and magnetic behavior together make nanoparticle significantly fascinating, but also raise important consider for the safe use in biology and medicine. Their physiological proper influence are shape and aggregation, surface chemistry and charges, composition and size. As I say, surface particle size and surface area is may major roles. Smaller nanoparticles have higher surface to volume ratio and increase the reactivity and potential interact with the cells and biomolecules. Shape and morphology influence uptake and distribution. From example like road shape or fiber-like particles such as carbon nanotubes can behave differently compared to spherical particles in terms of cellular uptakes and clearance. Surface chemistry and coating is very important. Modification like surface charges, functional group, impurity can affect solubility, protein binding, even toxicity. Another critical point is aggregation and agglomeration. Nanoparticles rarely exit isolated particles. They can often cluster together biological fluid, which can alter their effective size, mobility, and interaction. Finally, the solubility and resolution rate. So overall, physiochemical properties such as like size, shape, surface chemistry, solubility, aggregation, and strongly influence how nanoparticles behave, how they are disturbed in the body, and ultimately their biological and toxicology outcome. So next, what is the nanotoxicology? The nanotoxicology is the study of toxicity of engineered nanomaterials. It investigate how unique nanoscale property influence interact with the biological system, potential leading to adverse health and environment effect. This graph shows dose dependent toxicity. Very low dose human exposure rate, nanoparticles may not trigger significant toxicity. However, higher dose, multiple biological system get saturated. Metabolic enzyme saturation, enzyme can be no longer neutralized. or detoxified nanoparticles, saturation of binding sites on protein and DNA, abnormal signaling or mutagenic effect. Saturation of receptor transfer proteins alter the cellular uptake and trafficking. Saturation of defense and repair enzyme, oxidative stress, impaired repair lead to the toxicity. The key point is nanoparticle toxicity often non-linear. Small increase in dose can suddenly overwhelm defense system and lead to disproportionate effect. Nanoparticles have different types of frameworks, which like example source can be come from the intentional manufacturing, nanomedical, electronics, cosmetic, or unintentional like combustion, wear and tear. Exposure pathway by human ecosystem encountered by nanoparticles via air, water, soil and dermal contact. Receptor interaction one inside nanoparticles interact with the cell membrane, receptor organelles and biomolecules. Risk assessment on the basis of evaluated how exposure level and potential hazard to the human and ecosystem. Predictive models as we can approach to the QSR bioinformatics help the nanoparticle behavior and toxicity. As engineering nanomaterial synthesis, we have to do characterization of size, shape, surface chemistry, solubility, and this determine the biological effect. Also, the life cycle, what cradle to grave nanomaterial can release during the production, use, and disposal. Dispersion and transformation by the different types of release undergo aggregation, dissolution, chemical modification in air, water, food and soil or sediment. Exposure assessment on the basis of how nanoparticles are present in environment and how human are exposed. Risk characterization on the basis of combining exposure plus hazard data to estimate actual risk. Bioinformatics, again we can use predictive toxicity prediction on the toxicity value nanostructure activity relationship. Also on the basis of nanoparticle size and distribution, same material can behave very differently at nanoscale. 50% of volume or 50% of number distribution. By volume, larger particles dominate in the measurement, masking nanosize fraction. By number, nanoparticles appear more dominant due to their high count, even if they contribute less to overall mass. This highlights why nanoparticles cannot be assessed during the bulk material and their size and surface area control their behavior. Here there is a different types of what is the difference with nanoparticle versus larger particles? You can see here the ratio characteristic. Nanoparticles have very high surface to volume ratio that make them the more reactive, allow them to interact more strongly with the biomolecules. Larger particles have much lower ratio, making them less reactive. Agglomeration, nanoparticles tend to clump together depending on the medium and surface chemistry. These change their effective size and toxicity. Larger particles are less likely to agglomerate. Deposition and mechanism in respiratory tract. Nanoparticles move by diffusion and can deposit throughout their respiratory tract, including deep alveoli. Larger particles deposit mainly by sedimentation, impaction, interpretation, usually in upper airways. Protein and lipid absorption. Nanoparticles are strongly absorbs protein, lipid both in vitro and in vivo, forming protein corona that indicate biologically identity. Larger particles absorb fewer biomolecules and this process are less biologically significant. Translocation to the secondary organs. Clearance. Nanoparticles are most likely to translocate one beyond lung into the bloodstream to other organ. Larger particles generally do not translocate unless there are overload in the lung. Mucocellular clearance. For nanoparticles, clearance may be slower but still occur, and larger particles usually efficient via mucociliary escalator. Bialveolar macrophage, it for nanoparticles is poor, and for larger particle is efficient. For across lung epithelium, nanoparticles can penetrate lung epithelium and enter circulation. For larger particle, mainly under overload condition. Lymphatic and blood circulation. Nanoparticles both can enter in both lymphatic system and blood circulation. Larger particles require overload and translocate. Sensory neuron and uptake transport. Nanoparticles can travel sensory neuron, but in larger but not likely to this route. Cell entry uptake. Nanoparticle enter via multiple mechanism like glycerin mediated endocytosis, lipid trap, diffusion can enter to the mitochondria, disturbing energy production. Very small nanoparticles even penetrate the nucleus, rising concern to DNA interaction. Larger particles enter mostly phagocytosis in specialized immune system. They do not enter mitochondria or the nucleus. The effect of this on the secondary organs like brain, liver and spleen, uh, they can reach. Larger particles they cannot, express overload condition. Portal inventory, both nanoparticles and larger particles are local effect such as irritation and inflammation. Inflammation can be occur nanoparticles as much lower doses. Both induce inflammation, but nanoparticles do have lower doses. Oxidative stress, nanoparticle can generally reactive ROS, reactive oxygen species rapidly due to their hard surface reactivity. Larger particles can also oxidative stress but higher concentration. Activation on signaling pathway. Nanoparticles interact with the cellular signaling cascade, altering cell growth, survival, and immune response. And both genotoxicity and carcinogenicity. Nanoparticle more likely to cause DNA damage, mutation, chromosomal changes due to their ability to reach nucleus. Larger particles have some effect but less frequently. So nanoparticles have classification on the basis of geometry and chemistry. The main base of their shape, dots, wire and sheets. Chemistry, on the basis of chemistry, their metal, ceramic, carbon and polymer. So dots, teeny shape like silver gold nanoparticles growing quantum. Wires and tubes, metals, silver, gold, nanoparticles, iron, cobalt, nickel, magnetic nanoparticles, copper nanos, ink, uh, gold and platinum in 1D and 2D, silver nanoplates. Metals like silver, gold use antimicrobial coating. Ceramic like titanium oxide used in sunscreen and paint. Carbons like nanotubes, graphene used in electronics and advanced material. And polymeric are softer, often biodegradable, useful in medicine and drug delivery system. There we can also show the surface charges, static interaction and agglomeration. So primary particles are individual nanoparticles. You can see here nanoparticle. In dry powder there are primary can be agglomerate, aggregate and agglomerate aggregate. So agglomerate loosely bound by weak one-day force, aggregate strongly bound by chemical bond, and agglomeration aggregated mixed stated with the both. Liquid dispersion, nanoparticle interact with the water and ion forming electric double layer around each particle. If there is zeta potential is measured by surface changes, high zeta potential, stronger repulsion between the particles, stable suspension. Low zeta potential, weak repulsion, agglomeration. Hydrodynamic diameter describe describe the effective size of particle in suspension, including solution layer, not for the core. Static force can be stabilized particles by preventing close contact. Primary particle size, hydrodynamic parameters, zeta potential, double layer thickness, and static stabilization determine nanoparticle stability and toxicity. Their surface reactivity mechanism catalyze oxidation on carbon defect on edge sites. Nanoparticle with carbon surface can catalyze deactivation antioxidant like glutathione, and this causes oxidative stress in cells. Oxidative release and metal ion is anaerobic condition. Aerobic condition. Nanoparticle release metal ion. This ion interact with the oxygen and protein, producing reactive oxygen species. Reactive oxygen species can oxidize stress, cause oxidized stress, damaging DNA, proteins, and membrane. Also, we can see the photochemical and redox activity. Titanium dioxide nanoparticles, which can, when exposed to UV light, they generate electron hole pairs. This pair redox activity produce reactive oxygen species. This explains why titanium oxide nanoparticles are photoactive and can damage cell under the sunlight. Metal catalytes reduced in carbon nanotubes. Reduced ion catalytes can undergo redox cycling. Example, ferric react with the H2O2, producing hydroxy radicals OH, which can be extremely toxic. This contribute oxidative DNA damage and cytotoxicity. So, you can see in this slide, the nanoparticles do not exit the isolated entity that interact with the surrounding and leading to aggregation and stabilization. Surface charges, zeta potential, steric interaction, and chemical reactivity dictate stability in biological fluid, uptake by cells, toxic potential through ROS generation, ion release, and catalyte activity. This explains why two nanoparticles of the same composition but different modification can have different toxicological outcome. So, in vivo and vitro mechanism, there are nanoparticle toxicity. First, we can see the in vitro mechanism. In vitro mechanism, the first damage to the cell's wall and plasma membrane by interact with the directly to the cell membrane. Their small size insert into lipid bilateral, causing membrane thinning and pore formation, increased permeability leading to leakage ion and molecules, so resulting compromised cell integrity and cell death. Interacts with the electron transport and anaerobic respiration by can localize in mitochondria, disrupt in electron transport chain, also lead to reduced ATP production during cellular energy crisis, excessive generation of reactive oxygen species like carbon nanotubes or metal oxide nanotubes impair mitochondrial function. One of the most oxidative stress universal mechanism, they can production of ROS such superoxide, hydroxide and peroxide, they lead to ROS production and which leading to lipid peridoxin, protein oxidation and DNA damage. Some of the activation of the cell signaling pathway like nanoparticles can activate false signaling molecules, also state related pathway like MAPK, NF-kappa and P53. Also lead inflammation, apoptosis and uncontrolled cell proliferation. Fifth, perturbation on ion homeostasis. Certain nanoparticles disturb cellular ion balance. They alter calcium, sodium and potassium channel, causing abnormal signaling and cell dysfunction. Example, intracellular calcium can trigger apoptosis. Release of toxic ion in internalized nanoparticles, like example, metal-based nanoparticles may dissolve inside the acidic compartment. They can release, bind to protein enzyme, disturb metabolic process, and induce toxicity. Disrupting of lysosome membrane integrity. Once internalized, nanoparticles often end up lysosomes. Some of cause lysosome membrane permeability, releasing digestive enzyme into the cytoplasm. They can lead to apoptosis and necrosis. Incomplete uptake and frustration of frustrated phagocytosis. Large or fibrous nanoparticles, like example, long carbon nanotube abstos like structure in interface with the cytoskeletal function, like nanoparticle can bind actin filaments and microtubes. This disrupts cell shape, motility, and intracellular transport. Impaired cytoskeleton also affect the cell division. DNA and chromosome damage. Nanoparticle can enter the nucleus if small enough, less than 14 nanometers. Directly cause DNA damage via ROS. Its effect DNA strand break, chromosomal abrasion, and mutagenesis. This raise concern long-term carcinogenic potential. So, nanoparticles have toxicity via multi-level mechanism, membrane, mitochondria, lysosome, cytoskeleton, and genetic materials. This combined effect explain why nanotoxicology must consider not just dose but also particle size, shape, and chemistry. Then toxicity of nanoparticle defined as a oxidative stress to frequently generate.
Namaskar, sir.
So oxidative stress nanoparticles often generate reactive oxygen species that cause redox reaction and metal ion release, damage protein, lipid, and DNA. Also trigger inflammation. They also cause the cytotoxicity, disturb cell membrane and organelles. Metal ion release can interfere with the cellular transport and reduce cell viability. Mitochondria often lead to the apoptosis, genotoxicity. DNA damage observed in vitro and in vivo. For certain nanoparticles type, metal and metal oxide are common culprit. So, uh, also the causes apoptosis and necrosis. Apoptosis is a programmed cell death and necrosis is an uncontrolled cell death. Depending on their physiochemical property and doses, nanoparticles may co-induce programmed cell death or uncontrolled necrosis. Organelle dysfunction by mitochondrial depolarization, disturb mitochondrial membrane and potential, lysosomal rupture, like nanoparticles often accumulate inside the lysosome, endoplasmic reticular stress, which can be interfere the protein folding inside the endoparticular layer, also activate unfolded protein response. If prolonged, enteroparticular resistance straight promote apoptosis through stress signaling pathway. So organelle dysfunction is central mechanism of nanotoxicology as a mitochondria, lysosome, regulate cell allergy, degradation, and protein hemostasis. So nanoparticle can trigger cell death and disturb critical organelles. This process explains how much cytotoxic observed in nanotoxicology study. Understanding which pathway, apoptotic versus necrotic mitochondria versus ER stress, will help in predictive toxicology outcome in different nanomaterials. Also, the different types of toxic effect. Example of toxic effect, like example titanium oxide, which is used in sunscreen and cosmetic product because of UV blocking properties. They cause lung inflammation in animals. Also, the silver nanoparticles is antimicrobial, so genotoxicity, neurotoxicity, harmful to ecosystem. Carbon nanotubes is long, rigid, multi-walled carbon nanotubes linked to abstract like lung pathology and rodent study. And quantum dots, it is a calcium based quantum dots release uh, toxic uh, uh, cadmium based ion, cause liver damage and neurotoxicity. Regulatory and risk management, international oversight. There is a OECD guideline to risk assessment for manufactured nanoparticle framework are emerging. Labeling, registry and data collection are emerging. Cosmetic and consumer products scrutinized for the non-ingredient safety. Exposure limits, regulatory bodies evaluate workplace limit, special concern on inhalation hazard, carbon nanotubes and uh, TiO2 are criticals. There are these different types of challenges, like nanoparticles are rapidly evolving. Uh, uh, Engineering nanoparticles are limited epidemiological data, strong precautionary regulation, international harmonization of safety standard, and more investment in long-term safety study. So regulation is catching up, but not yet fully developed. While OECD and national agency provide guidance, uncertainty among the long-term health effect remain. High risk materials like carbon nanotubes, titanium oxide, and nanoparticles are strict monitoring. Overall regulatory focus on risk assessment, product register, workplace safety, and the closing knowledge gap. So, for the nanoparticles, dose metric and portal entry. So, dose traditionally toxicology measure exposure in terms of mass concentration, but nanoparticles not it up. So, for that, key dose metrics are surface area, numbers, concentration, mass concentration. Most surface area most relevant mass based on nanoparticles surface drive reactivity, number of concentration important since nanoparticles are extremely numerous even low mass. Example 1 mg of 20 nanometers particle contain billions of particles. Mass concentration skill used but not understand biological impact of nano-sized particle. Comprehensive dosimetry often consider all three mass, numbers and surface area to properly link exposure with the effect. Portal entry like inhalation, dermal routes, injection and oral routes. So dosimetry matters. Toxicology influenced by the particle number and surface area, just the mass. Multiple entry routes, inhalation, dermal, injection and injection, each pose unique risk depend on the particle properties and application consent. Proper risk assessment must match right dosimetry likely to the. So portal entry in vitro, in which there are different types of concepts or nanomaterial physiochemical properties. Example via size, surface, chemistry, hydrophobicity, redox activity, interact with the body compartment media, respiratory lining fluid, gastrointestinal fluid, plasma protein and mucous membrane. Different types of factors like protein and lipid absorption, formation of protein, corona to change the particle identity, biodispersion, by the how nanoparticles cross biological barrier and distribute to tissue. In disease stage, implant tissue, alter plasma absorption pattern may be changed, modify toxicity. So portal entry in vivo, skin, you can see the skin, dermal exposure via air, water, cloth and topical product. From the skin, nanoparticle reach lymphatic system, CNS, PNS and eliminated via the sweat and exploration. Respiratory tract, Major route of airborne nanoparticles, particle deposit nasal, tracheobronchial and alveolar region. From here, nanoparticle translocate into blood and further into liver, spleen, kidney or heart. Gastrointestinal tract via contaminated food, water, hand to mouth contact. Nanoparticle interact with the gut lining and enter blood and lymph and reach to liver. Injection via medical or drug delivery, direct delivery into blood bypass neutral barrier, enable target application, also barrier raise systematic toxicity risk. Translocation and distribution. Once blood nanoparticles interact with the platelet, monocytes and endothelial cells, they can distribute CNS, PNS, crossing the blood barrier via olfactory or systematic route, bone marrow affecting hematopoiesis, and heart, liver, kidney, spleen, placenta and muscle. So extradite plastering by clearance by using urine, nanoparticle size chemistry, urine, feces, sweat, exploitation, and breast milk. You can see the risk assessment, implication, entry and distribution via dose response relationships. Toxics is not only about dose but also where the nanoparticle travel and accumulate. Key metrics are surface area, volume number, concentration, regulatory measure like NOEL, LOL, basal BMD to set exposure limit. Uncertainty exists due to interspecies difference, chronic exposure, data gaps, and evolving nanomaterials. So nanoparticles enter body via skin inhalation, injection, or ingestion. Once bloodstream, they can reach almost every organ depending on size and surface chemistry. Their fat, accumulated versus clearance determine the long toxicity risk. So challenge in nanotoxicology assay, different types of challenge are there. Absorption, nanoparticle can bind dye, nutrient, cytokines, use assay giving misleading result and false positive negative results. Agglomeration issue, nanoparticles tend to cluster altering their size, behavior. Agglomerate may be accelerate toxicity or interfere with the genotoxicity test like comet assay. Lack of standardization, no initial testing protocol exists. Standardized reference material, dose range and exposure time need to be reliable comparison. Here safety consideration in nanomaterial design. Traditionally nanomaterial develop often prioritize function first, which safety evaluate later. The safer by design approach flip this. It is integrate toxicology consider early in the nanoparticle design. Balance functionality with the minimize health and environmental risk. Nanoparticle can absorb biomolecule non-specific causing unexpected toxicology. By carefully turning surface chemistry, we can limit unwanted absorption and make distribution more predictable. Use biodegradable material. Incorporating biodegradable polymer or coating PGLA, PEG ensure the nanomaterial breakdown into safe byproduct. This reduces persistence in the body and long-term accumulation in organ, widely applied in drug delivery and biomedical nanomaterials. Surface modification, functionalizing nanoparticles surface with the targeting lesion, ensuring delivery to specific tissue or cells. Naturally polymers reduce immune recognition, protein absorption and inflammation, resulting lower non-specific toxicity and improved therapeutic efficacy. Avoidance of hazard elements excluding toxic metal, example cadmium, cobalt, or ensuring encapsulation can significantly lower the risk profile of engineering nanoparticles. This reduces genotoxicity, oxidative stress and organ toxicity risk. Regulatory alignment, designing nanoparticles to meet existing safety safety guidelines, improve the translation success and reduce regulatory delay. So safer by design, smarter nanotechnology by controlling absorption, biodegradability, safer surface chemistry, material choice, regulatory compliance, we can create nanomaterial that are both effective and safe. This approach ensures sustainability, public trust and successful commercialization. Route of exposure or toxicokinetic. Inhalation is a main exposure route. Ultrafine particles deposit deep in the lung. They cross into circulation. They move into other tissue, include the brain. Cross into bloodstream, travel secondary organ into the liver, kidney, heart, even the brain via the systemic circulator or olfactory neurons. Long-term inhalation may cause chronic lung inflammation, fibrosis, even cancer, seen in with the titanium oxide and carbon nanotubes. Dermal contacts, nanoparticles may interact with the intact and damaged skin. On intact skin, penetration is usually limited, but small and lipophilic nanoparticles can still enter hair follicles or sweat gland. On damaged or inflamed skin, penetration increased dramatically. Local effect like irritation, oxidative stress and inflammation. Systematic effects to nanoparticle penetrate deeply and enter circulation. Injection. Nanoparticle may injected directly by food additive, packaging supplement, or indirectly swallowed by the inhaled particle clear from respiratory tract. Inside the gastrointestinal tract, nanoparticles interact with the blood fluid, microbiota and epithelial cell. They can also absorb into blood and lymph and transport to the organ like liver, spleen and kidney. Some nanoparticles persist, disturb gut microbiome balance and cause organ toxicity. Biomedical delivery, intestinal injection in medical applications such as nanocarrier in drug delivery, vaccines, along with the imaging agent. Direct injection bypass neutral barriers, delivering nanoparticles straight to the circulation. Precise targeting tissue, potentially for the systemic toxicity, immune activation and long-term activation if clearance is poor. Once inside, nanoparticle may agglomerate, may translocate across the membrane, absorb macromolecules like proteins, corona, distribute widely and acclimate. They might be even resist clearance. Route of exposure and target organ. Inhalation is primarily airborne nanoparticles. Already we taught in the previously slide. Injection through the blood or water, target GI, tract, liver and kidney. Thermal through the cosmetic or sunscreen. Skin penetrate varies. Via medical application, directly enter the bloodstream. Common target, lung, liver, kidney, brain, cardiovascular system are frequently affected. So, when nanoparticles are inhaled, size determine where they deposit in respiratory tracts. There are three mechanisms to control deposition: impaction, sedimentation and diffusion. And these respiratory tract deposition, three mechanisms: impaction, sedimentation and diffusion. This mechanism improves where particles stay in upper airway and tracheobronchial region or deep in alveoli. Impaction, larger particles have high inertia and cannot flow airflow chain easily. They deposit in impaction in upper airways, nasal passageway or pharynx. Typically trapped in mucus and cleared by the mucociliary escalator. Relevance: inhaled dust, pollen, large droplet. Sedimentation: medium-sized particles settle out in airstream by gravity. They deposit tracheobronchial region and cause local inflammation and irritation. Fine particles matter, PM 2.5. Diffusion: they are small they move randomly like Browning motion, allow them penetrate deep into the lungs and reach to the alveolar sacs. Once they can cross alveolar capillary barrier, enter bloodstream, translocate to secondary organ. Example, ultrafine combustion particle, engineered nanoparticles. Respiratory tract clearance and deposition of nanomaterials by different way. Alveolar macrophage are the first line of defense. They engulf inhaled nanoparticles, attempt to remove them. Very small nanoparticle may invade macrophage uptake. Long rigid nanostructure can cause frustrated phagocytosis leading chronic inflammation. Mucociliary escalator, large nanoparticles and accumulation often get trapped in airway mucus. Mucus is constantly moved upward to cilia and eventually reaching to throat region and being swallowed. Then effective clearance pathway but indirect redirect nanomaterials into gastrointestinal tract. Dissolution, soluble nanomaterials dissolve into the metal ion. They can also absorb clear through the systemic circulation. They can reduce particle burden in lung but still cause systemic toxicity. Translocation, some of the nanoparticles cross alveolar capillary barrier and turn into bloodstream. Usually less than 1% of inhaled dose translocate. Once in the blood, nanoparticles reach liver, kidney, even brain. So overall, together process help manage nanoparticle exposure in lungs. If clearance is insufficient or overwhelmed, nanoparticles can accumulate chronic and consequence chronic pulmonary inflammation, fibrosis, and systemic effect. Nanoparticle clear by macrophage, mucociliary transport, dissolution, and limited translocation. Insufficient clearance, accumulation, long-term lung disease like fibrosis and cancer. This explains why inhalation is most critical exposure route in nanotoxicology. Nanomaterials and brain, blood-brain barrier, BBB crossing. Nanomaterial can penetrate blood-brain barrier through the various mechanisms, especially less than 15 nanometer coated with the surfactant or ligand via endocytosis, transcytosis across the epithelial cell, absorption or reception mediated pathway. Once they accumulate into brain tissue, raising concern long-term neurotoxicity. Olfactory transport, direct entry to the nanoparticles into the nose can travel into olfactory nerve. They bypass the BB transport completely, and olfactory bulb and hippocampus area are important for memory and smell. BBB disruption. Some of the nanoparticles can increase BBB permeability even if they don't cross efficient themselves. These transients opening allow other toxic molecules and pathogens can enter brain. This BBB disruption is secondary risk management. Neuroinflammation, oxidative stress, microglia activation, and chronic neuroinflammation all are related to the ROS generation, damaged neuron, release inflammation cytokines, and the like neurodegenerative disease like Alzheimer's or Parkinson. Brain accumulation occurs in specific brain region, including hippocampus and olfactory bulb. Accumulation may alter brain signaling and behavior over the time. So nanoparticle reach brain by crossing BBB or via olfactory transport. They can cause neuroinflammation, BBB disruption, long-term accumulation by potential link to neurological disease. Highlight of brain as a critical target organ nanotoxicology. So elimination by nanomaterials by different pathway. Entry and initial deposition. Inhalation. Inhaled nanoparticles deposit in naropharyngeal mucosa, olfactory mucosa, alveolar region of the lung. From here, they can enter multiple biological pathway, blood, lymph, cerebrospinal fluid, even brain. Once in the blood, nanoparticles circulate widely and can distribute to secondary organs like liver, kidney, spleen. Entry into the lymphatic system allow the bypass liver initially, but eventually return to the circulation. Excretory route through the urine, if very small, via feces and hepatobiliary clearance. Translocation of brain by nasopharyngeal or olfactory mucosa can move along the olfactory nerve and reaching the olfactory bulb and trigeminal ganglion. They may also CSF by crossing cerebrospinal brain barrier via disturbing blood-brain barrier. Once CNS elimination is much slower compared to the lymph or blood. CSF pathway. CSF circulation may help in clearance of nanoparticles from brain. However, insufficient clearance lead to retention of accumulation, especially sensitive organ like hippocampus. So these are the key CSF, BPB and cerebrospinal fluid. Oral elimination by the respiratory tract by mucociliary escalator, swallow and excreted. Systemic clearance via kidney, urine or liver feces. Brain clearance slower dependent on CSF circulation. Here we can renal clearance. Renal clearance by kidney route. Nanoparticles smaller than this 6 nanometers are sufficient filtered by the kidney, excreted direct the urine, enhanced by the neutral or negative surface changes, high hydrophobicity, benefits is rapid clearance, reduced toxicity, hepatobiliary excretion, larger coated and nanoparticles taken by the liver cells, hepatocyte and Cooper cell, route excreted via bile, GI tract, feces common for the lipid base and pegylated nanoparticles. Phagocytic mononuclear clearance cleared by the macrophage in liver, spleen, lymph node. Concerned long-term retention may lead to chronic toxicity. Factor affecting clearance, surface charges, hydrophobicity influenced by immune recognition. Protein corona can be enhanced or mask opsonization, shape and aspects after the circulation and clearance. Biodegradability determine persistent or breakdown. Design strategy, biodegradable material, PLGA, chitosan, silk coating, pegylated to avoid immune uptake, stimulate response system triggered by pH, enzyme and redox. So, predictive toxicology and environment impact on nanomaterials. Predictive toxicology computational model use QSR model for machine learning to predict nanoparticle toxicity. Osmotic aspect, transcriptome, gene expression changes, protein alteration, reveal molecular mechanism of nanoparticle toxicity. Tiered testing strategy, stepwise approach, in vitro screening followed by the in vivo confirmation, benefit, reduce animal testing, 3R principle, enhance hazard classification, acid regulation decision, ecotox nanoparticles, environment release via industrial discharge, wastewater, sewage, sludge, product degradation, aquatic ecosystem as most vulnerable. Ecotoxicology effect by engineering nanoparticles, titanium oxide and silver nanoparticles, algae reduce the photosynthesis, invertebrate impair growth, mobility, fish like reproductive growth or growth issue, toxicity factor like size, solubility and surface coating, what we are already taught in previously slide. So predictive toxicology tools computational plus omic are essential for forecast of nanoparticle toxicity. Tiered testing strategy reduce the animal use, improve the hazard assessment. Nanoparticle release into the environment can harm aquatic ecosystem affecting algae, invertebrate and fish. Toxicity depend on the nanoparticle size, solubility, surface modification. Bioaccumulation and food chain transfer. A nanoparticle can accumulate in the organism if their uptake rate exceed elimination. Like key factor are surface charge and size, influence the cellular uptake, solubility, soluble nanoparticle release ion, increase the persistence, surface coating, may slow, accelerate clearance, accumulation occur in specific tissue, food chain transfer. Nanoparticles don't remain restricted to one species, move across the tropical level, algae, fish, shellfish or humans exposed via the seafood consumption. The process called is biomagnification. Concentration increase as you move up the food chain. Ecological and human health. Ecologically, algae reduce photosynthesis, disturbing oxygen balance. Invertebrate impair the reproduction, growth and mobility. Fish, liver damage, reduced reproductive toxicity and behavioral changes. Human health. Seafoods consumption may lead to long-term nanoparticle exposure. Persistent nanoparticle or toxic ion pose risk for kidney, liver and brain. Nanoparticles persist in sediment and aqueous system, become a continuously source of exposure, and so elimination rate increase the potential chronic accumulation and ecosystem level effect. So at the end, the conclusion is nanotoxicology offer an enormous potential from the medicine to energy to consumer product. However, nanotoxicology is critical to ensure safety. Nanoparticles behave very differently from the bulk material due to their size, surface chemistry and reactivity. We have seen some nanoparticles can enter body via inhalation, injection and dermal exposure or injection. Deposit and distributed to organs like lung, liver, kidney, even brain. Cause effects such as oxidative stress, inflammation, genotoxicity and neurotoxicity. Clearance mechanism exists, renal, hepatobiliary, immune, but also nanoparticles persist and accumulate, rising concern about the long-term effect. Environmentally, nanoparticles can bioaccumulate and transfer to the food chain, affecting ecosystem and potential returning to the human. Predictive toxicology tools shaped by design strategy are essential for developing nanomaterials and maximize benefit while minimize risk. And the last, nanoparticles must be advanced in hand with the nanotoxicology, ensuring innovation is guided by safety, sustainability and responsibility. Thank you.